androgens in males, stimulating spermatogenesis, but they also have some other functions as well, producing the secondary sex characteristics during puberty, again, the deepening of the voice, the muscle mass development that we see differently in males, the redistribution of fat, the strengths and the aggression that we associate with males as well is attributed from these androgens, including testosterone. Regulating sex drive, which is the urge to copulate, producing protein synthesis and erythrocyte production, which is providing more blood flow to develop bigger muscles or more skeletal muscle mass, and so on and so on. So those are some of the other functions of testosterone aside from their role in spermatogenesis or sperm production and development. Okay, let's take a 10-minute break here. Let's come back at 11:03, okay? All righty. Okay, so we are back. We're gonna finish up this packet. Let's see if there are any questions. If you've got any questions, go ahead and let me know now while I pull this up. Okay. Okay, so let's speak here about what actually happens to a sperm as it becomes mature or as it develops. So a sperm has 3 different parts. We've got the head, which has the chromosomes, as well as the acrosome. The acrosome is kind of the tip of the head, and the acrosome has the enzymes that are required for fertilization. So the enzymes that are needed to burrow through the layer of the egg, we'll talk about that when we get to the ovum, are held here in the acrosome of the head of the sperm. A mature sperm also has the midpiece, and the midpiece has the mitochondria. This is obviously producing the energy, the ATP, to provide the motility, to allow the sperm to move. Sperm have a very long journey, and they can be alive for anywhere from 2 days to I think 3 days is the max, but generally around 72 hours or so. And so they need energy to sustain themselves on this journey, and to be able to stay alive within the female reproductive tract when they're deposited. So the mitochondria that are in the midpiece provide that energy. And then finally the tail is this long, it's kind of a modification of cilia. It's really this ending projection that has this whip-like movement that is what is actually propelling the sperm along. Together with the peristalsis of the tube. So don't forget that we talked about the peristalsis of the smooth muscle that's lining the vas deferens and so on. But together with the whip-like motility of the tail, that's what actually guides and moves sperm along their journey. And eventually when it gets to the female tract as well. So we've got the head, the midpiece, and the tail. And this is what the sperm is acquiring along the process of development. So when a sperm is first released into the lumen of the seminiferous tubules, it doesn't have these components just yet. But as it makes its way to the different areas that we spoke about, the efferent ductule, the epididymis, and so on, it acquires these other components to become a mature developed sperm. Okay. Sorry. Okay. So let's talk about spermatogenesis here. Spermatogenesis is where we have the development of a spermatogonium, which is a single undeveloped sperm before it's released into the lumen of the seminiferous tubule. And this is where meiosis takes place. Now again, you should remember meiosis versus mitosis from some of your other introductory courses. But meiosis is the process of sex cell division, whereas mitosis is the division of our normal, ordinary somatic cells. So other cells of the body, like your foot cell, hand cells, neurons, liver cell, etc. Those cells undergo mitosis, which is just the standard division. But sex cells need to undergo a very unique division so that they can end up with a haploid number of chromosomes. So when we say haploid, we mean that they must have half the number of chromosomes because they will eventually be combined with an ovum that's also going to have some chromosomes as well. So if they both have full sets of chromosomes, then we're going to end up with a chromosomal anomaly in that embryo. And so to avoid that, the ovum must have a haploid number, so half the set of chromosomes, so essentially 23 chromosomes, whereas the sperm also have to have that haploid number as well. And that is the goal of meiosis when compared to mitosis. So meiosis has 2 rounds of division. The first round, we end up with 2 secondary spermatocytes. So we start off with our spermatogonia, which is our undifferentiated, undivided sex cell. It divides into 2. One of those sex cells will eventually undergo meiosis. The other one sticks around to undergo mitosis to produce more spermatogonia. So both of these cells will not become sperm. That's going to kind of deplete the number of sperm we have. One of those cells have to stick around, divide as normal, undergo mitosis to generate another spermatogonia so that we can always have spermatogonia to develop new sperm. The other one undergoes meiosis. The first division, or the primary division, it gives us 2 secondary spermatocytes, and the haploid number here is 2N. So again, 2 sets of chromosomes, which we don't want. And then it undergoes meiosis again, and here we divide that number and end up with 1N. So a haploid number, or half the number of chromosomes, ends up in each spermatid. And the spermatid is the undifferentiated, undeveloped sperm. So here again, it doesn't have the acrosome, the tail, and the other features that it requires. That will eventually be developed, but it does have the haploid number of cells, or 1N, which is half the number of chromosomes. Not cells, chromosomes, the haploid number of chromosomes. Okay, and as I was mentioning before, this development takes place along the tight junction in between 2 Sertoli cells. So from the basal compartment, and then the luminal compartment, and then eventually it makes its way into the lumen. So we have the spermatogonia, which become spermatocytes, which become spermatids. We have 4 spermatids per spermatogonia. And then eventually we get 4 spermatozoa, which are released into the lumen. Here, I'll remind you again, they're not quite mature, although we can see in this depiction that they have the acrosome, they have the head, the midpiece, the tail. They're not quite mature, and we're still going to call them spermatozoa here, and not sperm just yet. Okay. So spermatogenesis takes place from the basement membrane along the tight junction all the way to the lumen. Following mitosis, we have 1 spermatogonia staying around to produce more spermatogonia, and then the other spermatogonia actually undergoing the process of meiosis. We then eventually end up with spermatozoa released into the lumen of the seminiferous tubules. They're immotile for about 20 days. So again, they have their structures somewhat, but they're not fully functional. They're still in that developmental process for about 20 days. And as they move to the epididymis and move along those other structures that we talked about in that sequence of structures, making their way to the vas deferens, they will acquire their full functionality. They will acquire motility of that whip-like tail. They will acquire, the full development of the acrosome, the midpiece with mitochondria, etc. So they're going to move to the vas deferens via peristalsis, the contraction of that smooth muscle, and then they're going to stay in the vas deferens until this male ejaculates. So until we have the initiation of the sex response, or the sexual response in males, which includes the arousal, the ejaculation, and then finally the emission of these sperm, they're going to stay put as mature sperm in the vas deferens. Now, let's talk about the sexual response here in males. This is controlled by the autonomic nervous system. Again, not something that we voluntarily control. This also involves spinal reflexes, and then there are 3 phases to this process. There's erection, which is governed by the parasympathetic nervous system. There is emission, which is governed by the sympathetic nervous system. And then there's ejaculation, governed by the sympathetic nervous system as well. The stimuli here are going to be mechanoreceptors of the penis, as well as other erogenous zones. Erogenous zones being the groin region, the neck region, etc., the mammary glands or the breast region. And upon stimulation of mechanoreceptors in these areas, we end up starting off this response with erection. We can also have descending signals from higher centers in the brain, such as the hypothalamus, which is to say that thoughts or memories or any images in the brain can also trigger this sexual response as well. So it doesn't always have to be a physical stimulus. It can be a stimulus from the higher centers, such as images, memories, thoughts. So let's look at this flow chart, which kind of lays this out a little bit clearer here. So we start off with the mechanical stimulation or descending signals from the higher centers in the brain, such as the hypothalamus. They relay on to the spinal cord, which stimulates the sympathetic neurons to the penis, it stimulates the parasympathetic neurons to the penis and inhibits the sympathetic neurons to the penis. And that eventually begins the process off with erection. So remember we said erection was triggered by the parasympathetic division of the autonomic nervous system. That brings about swelling or dilation of the erectile tissue. The arterioles that supply these erectile tissue become engorged. They induce more blood flow, and that is also promoting relaxation of the smooth muscle here. When that happens, the increased blood flow, the increased blood volume, and blood pressure stimulates the erection of the penis, for one, but it also compresses the veins that are surrounding the penis. And that prevents the outflow of blood that's leaving the penis. So it's kind of trapping blood in the erectile tissue of the penis and preventing the penis from going flaccid again, okay? So this actually provides a positive feedback loop. So if you remember from way back many moons ago in Unit 1 of Semester 1, we talked about positive feedback loops, and we said that there weren't too many examples in the body. Here is one really good example of a positive feedback loop. So we're not only preventing blood from leaving the penis, but we're actually promoting more blood flow to the penis. And this happens again and again and again to sustain that erection, but then eventually we're going to need a negative feedback loop when it's time to kind of end this cycle when that erection is over, okay? So we're promoting more blood flow to sustain the erection, but we're also compressing the veins, kind of squeezing down those veins and prevent blood from leaving the penis, which just potentiates more and more of an erection. The second phase here is called emission. And emission is the response to the erection that has been stimulated from those mechanoreceptors and from that initial part of the process. This is governed by the sympathetic nervous system. So here we see a shift in the neural activity from parasympathetic, which governs erection, to now sympathetic, which governs the emission. This is where we have rhythmic contractions of the epididymis, of the vas deferens, of the ejaculatory duct. And if you remember, the ejaculatory duct is that combined, that shared opening between the seminal vesicles and the vas deferens that opens out into the urethra, leading out into the penis. So we have contractions of the smooth muscle that make up these structures, and that actually moves sperm from where it was being stored in the vas deferens. So remember we said that sperm kind of just sits in the vas deferens until this sexual response is elicited. So then we have the secretion of sperm from the seminal vesicles we have the secretion of sperm from the vas deferens out into the urethra, and we also have the addition of the glandular secretions from the seminal vesicles and the prostate as well. We talked about the purpose of these secretions, to lubricate the tract, to add the enzymes that are required, et cetera, et cetera. And when we add the fluid from these glands, the fluid from the seminal vesicles, fluid from the prostate, when we add that with this mature sperm, that entire concoction, that entire fluid is what we call semen. Okay, so the addition of sperm with the secretions from the seminal vesicle and prostate together, that fluid is called semen. And that semen is released into the urethra. Okay. And then the final phase of the sexual response is ejaculation. This is where we see the penis coming into play. This is also mediated by the sympathetic activity. And here we see contraction of the smooth muscle of the urethra this time. So not the smooth muscle of the other structures, the epididymis, the vas deferens, etc. Here it's the contraction of the smooth muscle, as well as some of the skeletal muscle that's found at the base of the penis to dump or to deposit the semen that is now that mix of substance into the female reproductive tract. We also see the stimulating closure of the urethral sphincter. This is to prevent urine from leaving the bladder and interrupting this flow or mixing with the semen. And this is why males cannot urinate and ejaculate at the same time. The sympathetic system here is closing that external urethral, that internal urethral sphincter, and preventing the bladder contents, which is urine, from being deposited into the urethra at this time. The result here is that we have movement of semen through the urethra and eventually out of the body, okay? And ideally being deposited into the vagina, the female reproductive tract. Okay, so that brings us to the